I'm talking to Robert Plomin this morning. It's a great honor because I've been an admirer of Robert for years, probably since I was in my late teens. We'll just start. Robert, will you start talking a little bit about yourself and explain your background, please, for people who haven't heard of you?
Sure. First of all, it's great to be here. I think we've got a great podcast going, and I look forward to our discussion. So my background is described in my book, Blueprint, but I'll give you the 2-minute version. That is, I grew up in inner city Chicago, and some of the facts I'll mention are kind of interesting in light of my research. And one is that I grew up in a home with no books. My parents didn't get past high school because it was the Depression days. And my sister didn't do well at school, but I loved school. You know, I read early. I went to the library and brought books home. So that's a topic we'll get onto later. Why me and not my sister? And then because I was in the Catholic school system in Chicago, they did a lot of testing. And so as a result of that, I got scholarships to go to an academy, a Catholic academy in Chicago called DePaul Academy. And then from there, I was just going to work. I was working full time all the way through high school. And I thought I'd just continue doing that. I was making a lot of money working at a research corporation for school business associates. And it was fun. And I was traveling around the country, even in high school, organizing conferences for them. But then I heard from the university, DePaul University, to the academy, and they said I could go to university for free. And what's more is they'd give me a stipend as well. So being an inner city boy, I thought that was a pretty good deal. And so I went to university taking courses just to fit in my work schedule. But I was in philosophy, and there's an interesting story how I left philosophy and went to psychology. But then I also had another bit of luck, and that was I had an advisor who said, aren't you going to go to graduate school? And I honestly had no idea what graduate school was. You know, no one in my family, 32 cousins, no one went to university even. And here I was supposed to go to graduate school. And I said, well, I'm going to graduate, so why do I have to go to graduate school? But he explained to me what graduate school was, helped me fill out applications. And then I got another scam as a stipend and fellowship, studentship to go to the University of Texas at Austin in their psychology department. And then another huge bit of luck, it was the only place in the world that had a course in behavioral genetics. And another thing that's relevant to my research is that I was completely floored by it. You know, I had never heard about genetics in psychology, and it blew me away. It was mostly animal studies then. But what fascinates me is why 39 other graduate students didn't become interested in it. So I then was going to stay at Texas, but I got another wonderful piece of luck, and that is I got an offer to go to the Institute for Behavioral Genetics at the University of Colorado at Boulder with a joint appointment in psychology. And that was, the perfect job. It was the only institute for behavioral genetics in the world at that time. So I feel like I've had a great deal of luck, and I was just in the right place at the right time. So we can talk some more about that later.
Now, when we're talking about genetics here, I just want to make it clear. So people remember genetics from school, and I remember learning about genetics in school. It was classical Mendelian genetics, but we're not really talking about that. We're talking about quantitative genetics. So will you just talk a little bit about the difference between both of them so people know?
It's such a crucial point. I'm glad you started with that, because it is true. Almost everyone learns about genetics from Mendel and his pea plants. But it's important to recognize he was studying single gene mutations in pea plants. So if you were a pea plant and you had this one recessive mutation, so you had a double copy of it, you would have seeds that are wrinkled, not the way a pea seed ought to look. And the thing about single gene mutations like that is they're hardwired and deterministic. So when you say genetic, people think of these single genes, and there's nothing you can do about it. It's hardwired, determined, and it leads to a fatalism in a way. But the critical thing to realize is that for complex traits like psychological traits, as well as common disorders like most of the medical burden in society, these traits and disorders are not due to a single gene. They're due to many, now we know thousands of tiny inherited DNA effects. And that changes everything. It goes from being hardwired to probabilistic. So when we say there's a heritability of 50%, it means half of the differences we see between people in a trait, like weight or personality or psychopathology, half of those differences we see in the population are due to inherited DNA differences. So the main point to make here is we are not talking about genetic in the Mendelian sense of single genes. Each of the genes we study works just like Mendel says they do, but we're talking about thousands of these little effects. And so it's a probabilistic thing. It's about genes influencing your behavior, not determining your behavior. It's such a critical point. I wouldn't mind belaboring that a bit more even.
So if we think of every psychological trait imaginable, like if we think of things like obviously cognitive ability, you could have personality, but even life outcomes like divorce rate or maybe like likelihood of criminal behavior, all of those things are under significant genetic influence. If you take something like height, it might be 80% heritable, and then divorce rate also seems to be 40% heritable. And people could say, like, how is it possible that divorce rate could be half as heritable as height? For some people, that would be unbelievable. So do you want to just talk a little bit about how almost everything, every outcome is not necessarily fully determined by genetics, but it's under a lot of genetic influence?
Yeah. That's another perfect introductory sort of question. When I was in graduate school in the early 1970s, in psychology, you don't see the word genetics. The textbooks at the time said schizophrenia was caused by what your mother did to you in the first few years of life. Psychology was completely dominated by what we call environmentalism. That is the view that you are what you learn. And so it was quite amazing to find wherever we looked that genetic influence is important. And so what's happened over those 50 years since I started is that psychology has really come around to recognize inherited DNA differences as they affect behavioral traits. And we've gone from thinking nothing is heritable to having the first law of behavioral genetics, which is everything is heritable. You cannot find any trait in psychology that's reliably measured in an adequately powered study that does not show genetic influence. So that even includes some weird things like you mentioned, divorce. People say, well, how can divorce be heritable? Divorce, either happens to you or it doesn't. But then stop and think about it. And that's what I'd like listeners to always do. Stop and say, what about genetics? So think about who gets divorced and who doesn't get divorced. Can't you think of anything there that might be genetic? It isn't like the act of going to court and getting a divorce certificate. What kind of people? And I know because it's me. I've been divorced twice. And, I think you can easily think of things like, and a weird thing no one has talked about is I think some of the things that make you at risk for getting divorced might be some of the things that make you attractive in the first place. Like kind of being a free spirit, risk-taking, joie de vivre, and everyone knows after a certain amount of time, some of that wears off, and more mature people might just roll with it and say, well, that's getting mature and getting older. But other people just say, well, life is short and I want more fun.
Yeah, that's well put. So, I guess it's probably hard to imagine how you can go from quantitative genetics to actually these life outcomes. But is it true in the sense that at the moment we can predict that they're connected, but we can't bridge it mechanistically? Like we don't actually know what psychological traits exactly are acting in the environment. So for instance, it could be things like you said, like maybe it could be risk-taking or excitement seeking, or it could be maybe impulsiveness or aspects of personality, but we can't bridge that mechanistically yet. We don't know the bridge between the genes and the outcome, do we?
Well, two things there. One is we don't know the genetic mechanisms, but that's because if there are thousands of genes and each of them has such little effect, it's going to be very difficult to go from genes to brain to behavior. In fact, we can't do that very well, even for single gene disorders. Like one of the first ones discovered was phenylketonuria, PKU. And that's a single gene recessive mutation that causes about 1% of the mentally institutionalized, cognitively disabled individuals in the 40s and 50s when this was discovered. We know that it's an error of metabolism. That means you can't metabolize phenylalanine, which you get in your food. You get it from dairy products and red meat, and especially from breast milk, the colostrum in breast milk. So what's very cool about that example, we don't really know how that works, but in the brain. But we do know if you then have a diet low in phenylalanine, you dodge the bullet and children grow up to have pretty much normal cognitive ability. So that's the first part. We don't know the genetic mechanism. The second part is when we say something like divorce shows 40% heritability, meaning 40% of the differences between people, that is, of the reasons why some people get divorced and others don't, 40% of those differences in liability to divorce are due to inherited DNA differences. We know you didn't inherit genes to get divorced. It's always something else more basic. And there have been studies showing that personality traits I mentioned, like risk-taking, are part of that. I mean, if you were very low in risk-taking, divorce is a hassle. I mean, and it's a big risk. And so, you're less likely to do it. But there's probably lots of other traits as well. No one studies joie de vivre, but I think that's part of it too. So maybe depression on the other side. If you're depressed, maybe you are more likely to get divorced. So there's some studies showing some of the personality, I don't know, mechanisms might be too strong, but correlates of divorce. But that's always true. When we say even height is 80%, 90% heritable, we don't really know what that means. You don't inherit height. You inherit all these physiological mechanisms underlying height. And there's probably thousands of them, incredibly complex. So it isn't just unique to psychology, this problem of not knowing how the thousands of genes, the intermediaries between these DNA differences and the outcomes, the very molar, gross sort of outcomes we study. And that's going to be centuries worth of research, I think, coming up. But for me, I just like the idea of understanding that a large part of what makes us different from one another is inherited DNA differences, which, again, I emphasize, is so striking from 50 years ago when absolutely nothing in psychology was thought to be heritable. So much so that you really drew a lot of flack and antipathy if you even suggested that genetics might be important.
Yeah, I just want to pick up on that point. Having recently gone through psychology university, why is there still an underemphasis of genetics and what we know about behavioral genetics? It's just touched on. There's no dedicated modules to it usually. And there's still a huge environmentalism, I guess, emphasized in it when you're learning psychology. And actually, you could throw evolutionary psychology in there as well. Why are they underrepresented? And they're still underrepresented, to be honest. Is that just a lingering effect of the dominant environmentalist kind of viewpoint of the 20th century? Or is it because most professors aren't trained in the research, or what's going on there?
Well, who was it that said, Asimov, that science progresses one funeral at a time? It's the old guys sort of have to die off. And I've always taken a long view on it, on having an impact. Friends of mine would say, no, you got to hit people over the head with a 2x4 and say, genetics is important, get over it. But in a way, I think that's counterproductive. In a way, I really always felt better to keep your head down, keep collecting the data, and eventually the data will pile up so much that it's very hard to deny it that genetics is important. And furthermore, rather than convincing people who are firmly believe everything's environmental, like the psychoanalysts, for example, I think it's better to let one funeral at a time come along so that the younger generation will say, what's all the fuss about? Certainly genetics has to be important. How can you think genetics isn't important? Look around you. Look at the differences in height. See how they run in families. It's just sort of, it gets to be common sense once you accept that we are not solely what we learn, that our inherited DNA makeup makes a difference. One more thing on that topic of you were saying you didn't get much genetics. I really am on a crusade to say any psychology department that does not teach genetics is really doing a disservice to their students, because genetics is the future. And I really mean that. The most high impact papers that are psychological or behavioral, more broadly, are genetics. Although there aren't a lot of genetic researchers, the per capita research money that goes to this research is way off the scale of what psychologists get. It's big science, large collaborative studies with hundreds of investigators. And it's the same techniques being used throughout the life sciences. It's not something peculiar to psychology and behavior. All of the life sciences, biology, medicine, everyone is on this bandwagon. So not to tell students about it is, I think, a real disservice.
I was actually just about to make a point similar to that. So you've so many well-known psychology findings that don't replicate. Like you have things like growth mindsets, you've learning styles, ego depletion, priming effects. Like none of them have strongly replicated. It feels like the public doesn't know about what's really core here or what we really know. Because again, the environment, as we'll get to in a minute, is hard to measure. And I don't know if we've done such a good job measuring the environment. And because we've measured genetics so well, it's disappointing that people aren't more aware of these findings. And maybe just to illustrate this, can you just talk about the evolution of the research here? Like talk about how these classical studies, which you obviously did, were designed in more recently. When we talk about thousands or maybe even tens of thousands of genetic variants, like how do we capture them? Because it's obviously done with modern research approaches. So will you just touch on how we know this?
Yeah, very good question again. And it goes back to a century ago. The twin method was first realized. There's two types of twins, identical and non-identical twins. And I'm sure most people know about them. And identical twins are called monozygotic because they're a single fertilized egg, a zygote, that then splits in the first few days of life so that you get clones. You get two zygotes or embryos that are identical genetically. So we know that in terms of DNA sequence even. The other type of twin, 2/3 of all twins, are non-identical twins, and they're like any brother and sister. They're separately fertilized eggs. Rather than having one egg get fertilized, women often have more than one egg drop down in menstruation, and two are fertilized because, there's millions of sperm, so they're happy to fertilize as many eggs that are around there. And so those fraternal twins are 50% similar genetically. So the neat thing about this method is that it's like a natural experiment in which you have two groups, both are raised in the same womb in the same family, and you can even study same sex because half of fraternal twins are same sex. Identical twins are always same sex. So if a trait is influenced genetically, like joie de vivre, which no one has studied, you'd have to predict, if your measure is reliable, that the identical twins will be more similar than the fraternal twins, or in the UK, they say non-identical twins. So that's the twin method. And there's an amazing meta-analysis of thousands of these studies across the sciences, And so this has been the workhorse of what we call quantitative genetics, where it's not going to identify which genes are involved, but it's asking the more basic question. Is it true that environmental influence, especially family environmental influences, account for all the variants in psychology? And two surprising findings there is, one, inherited DNA differences account for half of the differences in behavior. And then the second finding that we'll get into is that the environment works very differently from the way we thought it worked. What we're doing with the twin method and the corresponding social experiment called adoption method, where you take genetically related individuals adopted apart. And if genetics is important, those people should be similar, even though the kids didn't grow up in the same family with those, say, biological parents. So both of these methods with very different assumptions converge on the same conclusion that substantial genetic influence, on average 50% of the differences on any trait or disorder, are due to inherited DNA differences. So that's just a huge effect. And that's why genetic results replicate. We didn't have any right to think that genetics would account for 50% of the variants. Because in psychology, it's rare to explain 5% of the variants. Now, if genetics had only accounted for 5% of the variants, those century of research wouldn't have found much because the studies weren't that large. If heritability was 5%, you'd need samples in the thousands before you could detect it. But because it's so big, you really only need samples of 100 or a few hundred pairs of twins to show that effect. So that's what turned me on to behavioral genetics, is these results replicate. In fact, at the time of the replication crisis 10 years ago, I wrote a paper called The Top 10 Replicated Findings from Behavioral Genetics, mostly sticking it to these environmentalists who say, oh, there's this effect and that effect. And one after the other in rigorous replication trials were not replicating, including the ones you mentioned. In fact, some people have estimated that maybe half of the so-called classic findings in a psychology textbook won't replicate if we ever get around to trying to replicate them. And it's important in terms of, you were asking about sort of the history of acceptance of behavioral genetics. It was actually a benefit that people hated behavioral genetics. It was hard to get stuff funded. It was hard to get stuff published. You had to reach a standard of proof that was so much greater than these crappy psychology studies that were showing what people wanted to show. All you need is a 15-minute videotape of growth mindset, and kids will be, forever good, well-adjusted, and achieving well. So I think it was actually, that was one of the reasons why behavioral genetics results replicated. One, because the effects were so big, and two, because the standard of proof was so high. In fact, you almost had to replicate stuff before people believed it. So I'm very pleased that these huge findings, like the importance of genetics to begin with, replicate time and time again. They never fail to replicate. And that is really outstanding.
It is incredible. Now, I just want to underscore something here that people, if they're hearing this for the first time, will find it counterintuitive. But the reality is we know the shared environment, like the family environment, what you're exposed to every day, which you would think intuitively matters a lot for things like personality. It doesn't actually matter that much at all. And something that maybe blew my mind when I came across it, I think I actually came across it in Kevin Mitchell's book first, in 8-10 years ago, and then you subsequently wrote about it, is that maybe stochastic internal processes or like an internal environment is what actually makes some of the difference. So things like somatic mutations, which I guess are random, things that just crop up, changes in your DNA, or maybe just the way your neurons are wired. Because obviously, if we take identical twins that are raised in the same household, they have the same environment, they're still not clones. So what actually matters in the environment is really not what we think. Am I correct with that?
Yes. Again, perfect question for this phase of our discussion, because behavioral genetic designs like twin and adoption studies set out to disentangle nature and nurture. So things run in families like weight. It's perfectly reasonable to assume, as everybody did until a decade or two ago, that differences in why children or people in general, that why they differ in weight is due to nurture. It's due to the family environment, the shared family environment of where it makes sense. Your parents give you your food, and they give you they model lifestyle choices for exercise and that sort of thing. So the nurture assumption, as Judith Harris's famous book was called, in 2006, something like that, whatever. That's what she means by the nurture assumption, that it's all nurture. So these genetic designs were set up to separate, to ask, is family resemblance solely due to nurture, or is it influenced by the fact that family members share 50% of their genes, except for identical twins who share 100%? And the amazing finding that people still have trouble understanding is what runs in families is DNA. Inherited DNA differences accounts for all familial resemblance. Nurture doesn't get a look in. But heritability is 50%, which means the other 50% is not genetic. But it's not nurture. So that's a paper I'm working on now for Behavior Brain Science, a Target article called Not Nurture. Could environmental influences in psychology be stochastic? And that's exactly the point you were making. You could look for what makes two kids in a family different, like in body weight. What could it be? It could be one was ill and the other wasn't. Could be one has a particular sweet tooth or something like that. But try as we will to find environmental differences that explain differences between siblings growing up in the same family. After 30 years, we come up empty-handed. And this is for all sorts of personality traits, etc. So the first thing there is that nurture isn't important, which is just very hard for people to accept. where you think family environment is all, so that's hard to take. But secondly, this other 50% of the variance, we don't know what it is, because it's not nurture, which is about 99% of all the environmental studies that in psychology. It's not that. And we've been also trying for 30 years to find environmental things that make siblings in a family different. Like that Judith Harris book I mentioned, she was saying, well, you're just looking in the family. What about peers? So kids growing up in the same family have different peers. Maybe they account for these differences, say in psychopathology or even school achievement. But no, we can't find those variables. And so, although absence of evidence is not evidence of absence, we're coming to the point where we've got to take seriously what I called in my 1987 behavioral and brain science paper, what did I call it? The gloomy hypothesis, the gloomy prospect. That is, maybe these environmental influences are idiosyncratic, chance, or stochastic even. So that's a topic that interests me greatly now. And people have talked about it, Kevin Mitchell, but many people before him as well, that biologists are very interested in the idea of noise. If you think about the complexity of going from DNA sequence in one cell to the complexity of organisms like the brain, the most complex organ on earth, I find it amazing that heritability can be 50%, because that means that the DNA sequence you inherited in the first cell with which you began life is still sending out signal after all these decades and trillions of cells. It's not surprising heritability isn't 100%. It's just amazing, though, that it's even 50%. And so once you realize the complexity of going from genes to brain to behavior, it's pretty easy to think there must be some noise in the system. It's not hardwired by any means. So biologists are very keen on these processes. One that interests me is the one you mentioned, somatic mutations, because we do know, like a touchstone here is what makes identical twins different, as you said. They grow up in the same family. Doesn't mean they have exactly the same environment, but they certainly have the same nurture, as we've studied. They got the same parents. They're in the same social economic status in the same family. But what makes them different? And what's neat about identical twins is it's not genetic. Genetic does not make, they're the same for inherited DNA differences. But what they can have is somatic mutations that build up during life. And there's one study that shows that identical twins have about 5 somatic mutations. By that we mean it's not the inherited DNA. It could be mutations anywhere in your body that pop up. And they won't be transmitted to your offspring either unless they happen in your ovaries or testes. So I think that's neat, because it isn't inherited DNA, even though it involves DNA. It's what we would read as a non-shared environmental effect, because it makes identical twins growing up in the same family different from one another. So that's like that's an example of how, even if it's noise, and these mutations are noise in the sense that they're completely random, and that means you could never predict them. But even though they won't predict, we can still understand them. And it's kind of cool to think that these somatic mutations make a difference. And the second part of that story is mutations, somatic mutations, meaning by somatic, we mean not in the inherited germline. It wasn't like in the first cell with which you began life. It's subsequent to that. So these somatic mutations that occur in identical twins early in embryonic stages, can you see that they will then have all millions and trillions of cells with that same somatic mutation? So the earlier it happens, the more diffuse, pervasive the effect of those somatic mutations. So that's cool. It's still random, though, and we won't be able to predict. But I think that's an example of ways in which we can understand this somatic chance processes. And even though we can't use them to predict behavior. And that's why the subtitle of my book, Blueprint, is called How DNA Makes Us Who We Are. And that got a lot of flack because it sounds rather deterministic. But what I hope the book makes clear is we're not talking about determinism. But what I'm trying to say with that subtitle is that inherited DNA differences are the major systematic force making you who you are. The environment's important, but it's not the environment we thought was important. It's not nurture. It's this mysterious non-shared environment, which increasingly we're coming to think are these stochastic influences like noise in the system. And psychologists are starting to get on this, talking about poly-environmental influences. Maybe what it is just like genes. There's not one gene. There's not one major environmental factor like how much your parents love you, but maybe there's many, many tiny experiences, poly-environmental experiences. And with real-time recording, social media footprint, maybe you can get at some of that, but you have to study it, you have to show it, and you have to show that it makes a difference in siblings. I think this will be, so I guess the point I wanted to make is psychologists think about these as external, exogenous environmental factors, that you can study if you had a real-time video recording or biofit data on somebody. But we should take advantage of what biologists are studying, and that is endogenous differences as well, because they could also cause these differences between identical twins.
So I guess one way I think about it sometimes is like if you gave someone, a different person, a recipe for a cake that you use, the cake is not going to turn out the exact same, even if they follow every single measurement you gave them. So there's just an element of randomness that you can't predict. The outcome is going to be a bit different, even if the DNA is identical and the environment is very similar. And also, I think that people might overemphasize the potential effects of like a big life event in putting you on a certain trajectory. But like you said there, with perhaps like large scale recording of environmental interactions, it's probably like multiple environmental, like maybe thousands of micro environmental experiences together might cause a little bit of a change, but we probably don't have that data yet. And we don't design studies widely enough yet to actually capture that. So that's an interesting area. I think somebody referred to it as an environmentome, maybe?
Environmentome.
Is that one way?
Genome are the 3 billion base pairs of DNA, and the environome are all these little environmental factors with one huge difference. DNA is grounded in a 4-letter alphabet, A, C, T's, and G's, in a spiral staircase of DNA, and we know how it's transcribed, translated, inherited. And now that we're doing whole genome sequencing of all 3 billion base pairs of DNA, across the 23 chromosomes, which we call the genome, that's all she wrote. All of inherited differences have to ultimately be there because that's all there is. Whereas when we talk about environome, we don't even know, not only do we not know what it is, there's no simple code, like a 4-letter code for the environment. We don't know how it's transmitted. We don't know where it has an effect. We don't know anything. So that's why a paper I'm writing now is called Genetics is Easy, the Environment is Hard. Just making these very points. So that's a really, I hope people play back what you just said, because that's exactly right.
I think like if we've any chance of actually getting results there, we're going to have to use machine learning methods and obviously modern AI methods, because obviously traditional approaches won't possibly account for that, because it's going to have to be predictive, isn't it? Like you're going to have to aggregate so many different examples and individuals and across populations. It's going to be a hugely difficult project, and you mightn't even find anything.
Yeah. Well, I think you might not find anything because although this is a little esoteric, what does random mean? Well, it's important to mention this. A famous article by Thomas Eagle in 2005, who's a famous philosopher of science. The title of his article is "Random is Unpredictable." So he defines random operationally as unpredictable. And so this idea of poly-environmental influences, at some point, there are going to be such little differences, they might as well be they're going to be unpredictable. They're so small. Not to say you shouldn't study them. Fine. I think people let 1,000 flowers bloom and let everybody study everything. But by stochastic, well, first of all, with random, we don't mean luck or chance. So if you think of a roulette wheel, chance is the probability that your ball will fall on green or black or red, that is. So it would be 50-50 if they were divided evenly, but there's one space that goes to the house, which is where the house makes all its money, because you don't actually have a 50-50 chance of winning on that. That's chance, probability of an event. Luck is dumb. Luck is like superstitious behavior. And you see it on the roulette table across the world. Number 7 is far and away more often chosen, even though 7 has absolutely no greater chance of coming up than anything else. So luck is just dumb. It's like superstitious behavior. But now stochastic is an important distinction, at least conceptually. And this paper I'm working on emphasizes stochasticity. I don't know if it's a mistake, but random means you can't predict. Stochastic means you can never predict. That is, even if you found every single little thing, even if every atom you'd know, and you replay the tape, you'll get a different result. And that's stochastic. So it's fundamentally unpredictable, which is an important distinction, because with the randomness and, unpredictability, there's always like a factor X. Well, if we just study it deeper, we'll find the causal predictive determinants of behavior. And I think the distinction is important, because I think it's important for scientists to give up on their reductionistic, deterministic, mechanistic view. If only we knew enough, we could predict everything. And I think we need to be more humble about the fact that we can't predict that much. I think it's amazing now that with DNA alone, we can predict 15% of the variance in school achievement. And some people say, oh, it's only 15%. And I would say, show me anything else that predicts better. And we're never going to predict 100% because it's only 60% heritable school achievement. So that's a ceiling on DNA prediction. But I think it is an important view that we need to move away from a complete deterministic view. And that's happening across science now. In cosmology, it's fundamental to quantum mechanics, isn't it? Quantum uncertainty. And all the way out to cosmology, people are really thinking it's really chance in terms of dark matter and whether the universe is expanding or retracting, or, so, and also there are books on chance, randomness in sports, in gerontology, economics. That's why economists need a big dose of this humility, because they really think you can predict everything. But what we're seeing is they can't predict much at all. So I do think it's an important topic in science, but also in society. Maybe we can get into this. Parents don't have nearly as much control over their kids' development as they think. Genetics is important. That's systematic predictive variance. But in terms of what the parent can do environmentally, they don't have much effect. And it's partly because of what we're talking about here. And then thirdly, I think it's important in terms of understanding ourselves. I'm getting more and more interested in Buddhism, which amazes me because I'm completely an empiricist about things. But I think the idea of impermanence, a lot of the basic Buddhist principles are good principles to lead your life by. And I'm especially feeling this now because I just got back from 2 weeks in Bhutan, where the people, that's next to Nepal and Tibet and China and India, and they've been isolated for centuries, and they really live Buddhism. I'm not spiritual or religious at all, but they believe compassion is very important, recognizing the impermanence. Things change. What appears disappears. So I won't go on about it, but I do think this idea that it's not all determined is important in science and in society, especially for parents, and then also in terms of understanding ourselves, which leads me to, I'll say 2 more things while I'm on this rant. But one is we might have to talk about Robert Sapolsky's book, Determined, because, that's saying everything's determined. And I'm saying, even I'm saying nothing's determined in the sense that genes don't determine behavior. They influence. It's an important distinction, I think. But then, anyway, so we could maybe go into Sapolsky later. I really disagree fundamentally with what he's saying.
I was going to mention for a different reason to do with epigenetics and another book he wrote, but yeah, we can mention him in a bit. I just really want to bring up though, before we get to anything else, we know that psychiatry and we know that clinical psychology has obviously underwent kind of a crisis of confidence because of these categorical disorders. Now, I think I came across for the first time 10 years ago in your book first, in Blueprint, that we can't really categorize these as disorders in like a classical medical sense with like this checklist of symptoms and you have depression. Obviously, comorbidity presents a massive like undermining of that. So can you talk about like, is there really disorders? Because I think you probably on the side there's not, and these are really quantitative traits. Like, so I think the phrase used is abnormal is normal, that everybody has obviously these traits or the genes maybe that predict these traits, and they're dimensional. So maybe if you have them in higher concentration, that you will have some dysfunction, but like, the problem is that you can have dysfunction across different disorders. So just because you have some depressive symptoms, you don't necessarily fit the map of clinical depression. So I think that's obviously held back treatment, and it's held back like public understanding, because you see now on social media, people obviously claim that they have ADHD, and they probably do have an ADHD diagnosis, but maybe they're misunderstanding what's going on there. And it might be good for reducing stigma, but it probably isn't giving. See, what happens is people push back and they say there's this crisis of overdiagnosis. But the problem with the overdiagnosis thing as well is the diagnoses in the first place don't make sense. Like, again, you can have somebody who's below a clinical threshold who does have struggles, and it's understandable since these traits are distributed across the population normally.
Yeah. Well said. This is a very big topic. It's a newer topic and still somewhat controversial, but I have strong views on it. I wrote a paper in 2009 called Common Disorders Are Quantitative Traits. So it's exactly what you're saying, is we fool ourselves in thinking that even common medical disorders like high blood pressure or any of the psychopathology disorders are truly disorders in the sense of etaologically distinct. Now, the reason it's always been assumed that's the case is the medical model. So if you take, say, COVID, for example, you only get COVID if you have the SARS virus. Now, you might get sick for lots of other reasons, but it's a single cause. And similarly with single gene mutations, it's a single cause. Now, when you have a single cause like COVID, you notice all these people getting very ill all of a sudden. What causes it? So the first thing you want to do is diagnose it carefully to say, well, does this person really have it or not? And you get a group of people that really have it, which was pretty obvious in the case of COVID. Then you say, what's the cause? And you find the cause. It's SARS. It's nothing else. It's one single factor. Now, that the medical model works brilliantly, like it did at first with cholera and contaminated water, a single cause. If you got a single cause, the medical model is absolutely the way to go. The problem is with common disorders in medicine and psychiatry, it's not at all that way. There's nothing that's caused by one single thing. Genetics, we know now, is caused by thousands of little differences. And environment, despite people trying to find these big factors like parenting, it really doesn't pan out. So there's solid evidence, though. It isn't just, argument. It's solid evidence that disorders are dimensions. And now with DNA, the DNA revolution, which I hope we'll talk about, we now can say, here's a few thousand DNA differences, inherited DNA differences, that can predict maybe 7-8% of the liability to schizophrenia. Well, those polygenic influences are distributed normally. There's no breakpoint. There's no genes for schizophrenia. We all have thousands of schizophrenia genetic risk factors. It's completely quantitative. It's a matter of more or less, not either or. And that is an important distinction. You said in terms of stigma, for example, it isn't like us normal people and those schizophrenics. We all have thousands of these differences. And it's important to emphasize, we're not saying there aren't problems. People, if you meet schizophrenics, they really are not happy campers. You wouldn't want to wish schizophrenia on anybody, despite what people say about neurodivergence and all of that. I mean, this is not a happy way of living your life, out of touch with reality completely. So there are these problems, but what's to be gained by accepting the medical model and assuming that schizophrenia is etiologically distinct? That is, there are environmental factors like parental abuse, or it used to be schizophrenogenic mothers. How about that one? It was all mother blaming, basically. Well, that's not true. But what about genetics? There are no genes for schizophrenia. I mean, there's always a few exceptions. There's some very rare mutations that can cause something like schizophrenia. But schizophrenia out there in the population as it gets diagnosed is not etiologically distinct, genetically or environmentally. And there's a lot of important issues that follow from that. And that's only the first point here about what's wrong with our current diagnostic schemes. But the second one is the one you said, comorbidity. But before I go into that, do you want to say anything more about disorders are dimensions?
Let's just actually emphasize maybe how we know this, because I think people probably assume that it's like something goes wrong genetically, and that's why you get schizophrenia, or that's why you get a severe mental disorder. But we know, since the entire human genome was sequenced, and maybe we should mention GWAS here and polygenic scores, and maybe emphasize how it works. That's not really what's going on. It's not like they're not single gene disorders. We know that because anyone who's ever tried to test single gene influence or candidate gene studies where they just isolate one gene, they don't work out. So should we just emphasize how we know this, like GWAS and polygenic scores, and maybe the role of SNPs, like single nucleotide polymorphisms here as well?
Okay, well, this gets us into the DNA revolution, which provides the proof positive of what we're talking about here, about dimensionality and comorbidity between disorders. But before that, we knew from quantitative genetics, you can study you can take a quantitative trait like depressive symptoms, and then you can study people who got diagnosed as schizophrenic. And so some cases, identical twins, one's diagnosed as schizophrenic and the other isn't, because it's only, say, 50% heritable. So you can say, but if we measure it quantitatively, we can see we call it genetic correlation. To what extent do the same genes that affect depressive symptoms affect this diagnosis of schizophrenia? And the answer is a lot. So this has been done for most common disorders like ADHD or whatever. You can do a twin study, not just of diagnoses of depression, but of the relationship between a diagnosis of depression and quantitative symptom counts for depression. I know it's a little hard to understand, but that really paved the way. Whenever we look at it, we find dimensions. Disorders are dimensions, genetically at least. It's the same thing. But the proof of the pudding, as you say, is with DNA, the DNA revolution, and as you also say, candidate gene approaches, where early on it was expensive and time-consuming to measure one single DNA variant. And so people studied small samples, and they just studied a handful of genes. If you're interested in depression, people would study serotonin and serotonin transporter genes because they seem to make sense. They're good candidates. And what you didn't actually say is how many thousands of these studies were reported and how they completely washed out. When people replicated them with more powerful studies, larger samples, they don't work. And this was genetics' contribution to the replication crisis, because they did everything wrong that we talk about with the replication crisis. So small samples and chasing p-values, the whole schmear. But when that changed in the mid 2007, when people began to, two things. One is there was a development called a SNP chip, which is an array the size of a postage stamp that can genotype hundreds of thousands of DNA differences of a particular kind. We call them SNPs, single nucleotide polymorphisms, a difference in one step in the spiral staircase of DNA. So there's 3 billion steps. 99 point something percent of those are identical for all of us. But say something less than 1% of those steps differ between us. And when we talk about inherited DNA differences, that's what we mean. And the difference is now we can measure them, but instead of having to do one at a time, these millions of SNPs that each of us have, these SNP chips, as they're called, can measure them cheaply, like 50 quid or so, and inexpensively and quickly and very accurately. So that's what's changed everything, this technological advance that allows us to study not just a few candidate genes, but genes across the genome. And we now have SNP chips that pick SNPs across the genome that best capture genetic variation throughout the 3 billion base pairs of DNA. I can go into that some more. It's a technical thing. But the important point is we can now measure hundreds of thousands of SNPs across the genome, and that's led to the opposite of a candidate gene approach called genome-wide association, which is an atheoretical approach. Rather than guessing which genes are important, which is a losing proposition, you can assess genes across the genome and say, are there any of these DNA differences that make a difference in a particular behavior that you study? And that's when we do that, you find you can get replicated differences, but the biggest effects are so much smaller than anyone ever thought. So that gets into that polygenic issue we were talking about early. It's proof positive that we're talking about heritability of all complex traits and common disorders being due not to a handful of genes, but to tiny effects of thousands of DNA differences. And there's a lot that follows from that. One is, it's not going to be an easy ride to go from genes to brain to behavior if there's thousands of genes affecting these traits, each of which has a very small effect. The other side of this is that, as psychologists know, if you have a very small effect, you need a very large sample to detect it. And so GWAS studies, the first one was this Wellcome Trust study of 7 common disorders, one of which was bipolar, with 2,000 cases versus controls. So you're looking for DNA SNP differences between your cases and your controls. And people thought at the time, 2,000 cases, that's just ridiculously large. But from that study, we learned the biggest effects are very small, and you could just barely detect them with sample sizes of 2,000. The more recent studies of bipolar have not just 2,000 cases, not just 20,000, but we're getting on 200,000 cases, so that you can pick up smaller and smaller effects, and then you can put them together. So what good are these tiny effects? That's what you were mentioning before, polygenic scores. You can add these up because you can add up each tiny effect weighted by its effect size. So a SNP that's more correlated with the trait, you want to count it for more. It's just what we do when we take items on a questionnaire, personality questionnaire, say. We don't have one item, we have a bunch of items because we want to capture the construct better. So we add up the items weighted by, say, their reliability or their factor loading. So it's the same sort of thing. And it's these polygenic scores that are really changing science and especially psychology, because you need these huge samples to do a genome-wide association study. But once you get a polygenic score, you can just use a polygenic score in samples of 50 or 100 because the effect size isn't tiny. It gets to be pretty large. As I said, 15% of the variance of school achievement. So it's now the case that if you do a large study, you put in a grant proposal for a large study in psychology or psychiatry, if you don't have a chance of funding if you don't put in for DNA, because DNA is cheaper than a lot of the stuff we do. You say $50 all in, and even for whole genome sequencing, the price is down to, say, the real costs are on the order of $100, you know? So you think of fMRI for one subject is $450. I mean, you know, it's cheap. Plus you do it once and you get all the inherited DNA information that you can then use to study anything. So that's why studies of CBT versus drugs for depression, treatment studies, they have to include DNA because it's so cheap and it allows you to add a genetic dimension to what you're studying. It wouldn't just be on average in the population, is CBT better than drugs for depression? Instead, you can say, are drugs and which drugs useful for particular people based on their genetics? And you can say, you know, like my hypothesis would be people with high polygenic scores for depression will be more likely to be treated successfully with drugs. It's just a hypothesis, but I think of it as, you know, maybe more biological. So anyway, that's a lot to take on board. But the point you wanted to make was that these polygenic scores are always, for everything, perfectly normally distributed. And that's because of the central limit theorem of statistics. It's like, you know, you flip coins 10 times and you say, how many heads do you get? And the more you flip the coins, the closer you get to a normal distribution. You know, you can still get, if you flip it 10 times, you can still get 10 heads or 10 tails. But most of the time you get 5 heads and 5 tails and a perfectly normal bell-shaped curve around that. And that's what you're doing with SNPs. You've got 2 alleles, 2 forms of each SNP. And so that's like flipping a coin. But then you're flipping thousands of coins at the same time, and you always get a perfectly normal distribution with no evidence of a break point where we diagnose schizophrenia. I think it's kind of a scandal along these lines that proves that we're talking about genetics being perfectly dimensional, no evidence whatsoever for categorical disorders. And so I think with developmental disorders like ADHD and autism, these used to be very rare disorders. Now suddenly they're coming up to 2-3% for autism, 5% for ADHD. And I think it's merely, not a scam exactly, but it's just to say, if it's very rare, it's not going to help you very much in your office, in your clinic. But you can't have it so common that everybody has it because then it's not a disorder, clearly. And so they come up with 5%, 1 out of 20. That sounds like a real problem, you know, especially when you start saying, oh, look, we're the increased prevalence of these disorders. What's going on in our society? And, you know, I just look at it, it's all Alice in Wonderland stuff to me. I say, on what basis do you think there is a disorder? And when I first started saying this 20 years ago, when I came to the Institute of Psychiatry at King's College London, which is, you know, kind of the top place for psychiatric research, I thought people would have a fit when I started talking about it. Not at all. Any clinician working with alcoholics, obese patients, schizophrenics, you don't wake up one day alcoholic or obese or schizophrenic. You know, it's a process. And with schizophrenia, it's often, it's the functional aspect of it. I mean, you could be pretty weird. Like my supervisor, PhD supervisor at the University of Texas, who shall be unnamed, most people said would be locked up if he weren't so bright. I mean, he was really difficult. But you know, there are compensating factors there. I mean, he was extremely bright. And so it's not just yes or no schizophrenic, and it's not those schizophrenics out there versus us normal people. I think there's a lot that follows from this in terms of our own lives, but certainly from the science of it. And I would just conclude that by saying I can think of no reason scientifically why we should accept the medical model that disorders are etiologically distinct. There's nothing to be gained and an awful lot to be lost because we're doing our GWASs, millions and millions, probably billions of dollars now, on zeros and ones, cases and controls. And it just seems ludicrous to me because if you get quantitative data, which you can get, you know, there are people doing symptom, quantitative versions of these measures. If you do a quantitative thing, you can cut it off at a point and call it qualitative. You can say, okay, people above this line we'll call disorder. But if you only collect zeros and ones, you can't do quantitative work. So it seems to me we're missing a tremendous amount of information, because we call these people, psychiatrists call these polygenic scores polygenic risk scores. They think it's a risk for, say, ADHD, for example. But it's not. It's a normal distribution. It's as much predictive of low ADHD symptoms as it is of high ADHD symptoms. And I think it's important that we use all that information to help us find the best polygenic score predictors of these problematic dimensions first. And the second thing that I find very interesting is they're not risk scores. They're not just scores for high ADHD. They're scores for low ADHD genetically, the low end of the distribution. And what is that? Is that just low risk for ADHD? I really don't think so. If ADHD is primarily attentional problems, say, the other end of it is not just good attention. It could actually be compulsiveness, too much focus. With BMI, body mass index, a high genetic score, and that's my highest polygenic score, is a risk for, they called it obesity, but I would say high BMI, for example. But what's at the low end? Is it just low risk for being obese, or is it something else? And I really think if we studied people at that low end, we'd find that's where a lot of eating disorders are, where you become easily repulsed by food. I think we could induce dislike of red meat in people. I mean, with a lot of images of bloody meat, and, you could make it disgusting. And I think we're evolved to be disgusted with certain foods because that was adaptive. If something isn't quite right, smells bad or whatever, you get turned off to it. So I think there's a lot to be gained here by thinking about these things dimensionally, scientifically, especially interests me now. And again, nothing to be gained by pretending that these are ideologically distinct disorders. And I would say, just for people out there who, I know a lot of people get upset with this, because a lot of people, like I have a 45-year-old friend who called me a month or two ago saying he realizes why his life is fucked. He's gotten a diagnosis of ADHD. Now, it makes him feel a lot better, but he's still got all these problems. He can't concentrate. He's just, a flibberty gibbet, we used to say, just flying all over the place, low attention span, which makes him a really interesting character. But it's hard for him to focus and get things done. So I know increasingly now people from your generation or younger everyone has a disorder now. The more specific, the better. And, bless them if it makes them feel better, but it sure isn't helping them to understand themselves. In fact, it's counterproductive to understanding themselves. They got to say, yeah, I've got some problems, but it doesn't mean, even if there's genetic influence, ADHD is one of the more heritable disorders in childhood. It doesn't mean you're destined to be ADHD. Like my high polygenic score for weight doesn't mean I'm destined to be fat. In fact, it's been very motivating for me to fight that tendency, to gain weight easily and find it hard to lose weight. So that's another long-winded answer to your question about the dimensionality and how we know it. But I hope people get the message that this isn't just speculation. This is really true, that these liabilities are dimensional, not dichotomous or categorical.
I just want to mention as well, so what I find irritating about the whole thing about the overdiagnosis and the categorical diagnosis is sometimes if you say you get a diagnosis of depression or an anxiety disorder and you might go to the GP, like the diagnosis, as you said, might make you feel better, but then you get this blanket treatment of you get whatever SSRI is most common. But my issue is that just seems not sophisticated at all. Obviously, certain people respond to things differently. So they try one SSRI and it doesn't work and they give up and they get horrible side effects. But it would be better if we could predict how someone will respond to a given medication, because there's obviously lots of different medications and people respond differently to all of them. And maybe you need something like an atypical antidepressant, or you need a certain type of behavioral therapy or behavioral intervention, but it's kind of blanket treatment. And because obviously GPs and doctors view them as categorical disorders and don't know much beyond that, it's kind of limiting treatment, and people get dissatisfied with medication and dissatisfied with treatment. And people say things like, pills are dangerous, or they just give you side effects. And it's kind of frustrating, because I feel like, as in general, public and maybe even some people who actually work in the medical community don't fully understand what's going on here.
Yeah, that's another very good point. It's a huge area of research, drug by treatment interactions, and finding genes to do exactly what you said, to predict which depressant drug you'll try. Because on average, you don't just try it one night. You got to do it for weeks to see if a particular SSRI works. And then if it doesn't, you go on to another. On average, I think severely depressed people diagnosed as having major depressive disorder go through almost 3 drugs before they hit one, and a third maybe never do find one that works. So all of medicine is shifting now from the idea of waiting until people have a disorder and then trying to cure it, like having a heart attack and you go to the hospital and they try to patch you up, to moving towards a model of predicting and preventing disorders. It's in every way, socially, economically, ethically, has to be a much better thing to do. And DNA is the best early warning system we have. And that's why in the UK, at least, I don't know about in Ireland, that they're now really moving heavily. Wes Streeting, the current head of cabinet minister for health, is very keen on this. Because, if you can predict who's going to have a heart attack, a severe heart attack early in life. And again, it's probabilistic. But if there's 5% of men in the UK walking around with like a 10-fold greater risk of a severe heart attack in early midlife, 40s, say, that's quite rare. And if you knew you were one of those guys, it doesn't mean you're absolutely going to die of a heart attack in your 40s. But you sure as hell would pay a lot more attention to the messages we all get about eating well, staying healthy, keeping your weight down, and perhaps checking. You can go higher technologically than that. There are whole body scans now that can detect fuzzy arteries before they block up and cause a heart attack. And it's said that if you can prevent one of those heart attacks, you could save the NHS 700,000 pounds, let alone quality of life and everything else. So it does seem like a no-brainer, which is why medicine across the world is really moving in this direction.
I just want to jump in there as you're saying that. So one thing that I'm interested in is embryo selection. Obviously, this has been done for a while, but it seems there's some companies in the US, and you probably know this, that are starting looking at these polygenic traits or these behavioral traits. I'm early days here. I've only read a little bit on it. I read an article by a guy called Scott Alexander, who's a very well-known blogger online. He's a very reputable source. I read it on Substack, but I just want to get your views on that. Where are we now with it, and how good is it going to get? I know it's expensive, but eventually the price, we know the price of DNA prediction came down a lot. So I imagine the price will come down. So where are we now, and where is it going? And do you think it'll become something that a lot of people do?
Well, this triggers another very long discussion because there's so many issues involved. It begins with the DNA revolution, so we can now predict using DNA alone. But first, I'd like people to know that we've been testing neonates at birth around the world since the 40s and 50s. They've been tested for a few gene mutations, one of them being that PKU I mentioned. Because if you could detect at birth which kids have a double dose of that recessive mutation for phenylketonuria, you can then give them there's a simple intervention, a diet low in phenylalanine. You cut out dairy, you cut out red meat until the brain develops into adolescence at least, so that you dodge the bullet. And it's not fun to eat a diet low in phenylalanine, but it's a lot better than being very cognitively disabled and institutionalized. And if you don't do that, you're going to be stuck with someone for the rest of their lives, and they live a pretty full life, are going to be institutionalized. So that's an example of, you've got to do it. And so in the UK, we've tested for about 10 or 12 of these single genes for decades. But just last year, they announced that they're now going to be testing babies for 250 single gene mutations that are actionable. But now they've got the DNA, and it seems to me impossible that they will not look at some of these polygenic scores. I mean, the single gene mutations are important because they are hardwired and deterministic. If you have that mutation for Huntington's disease, like Woody Guthrie in that current Dylan, wonderful film about Bob Dylan, called Rolling Stone, what is that called? A Complete Unknown. The film, it's a wonderful film. And Woody Guthrie is in there because the only person in the world Bob Dylan was nice to was Woody Guthrie, who was in advanced stages of Huntington's disease. So if you were Woody's son, Arlo Guthrie, also a famous folk singer, you'd have a 50% chance of having it because unlike most single gene disorders, that's a dominant one. So anyway, if you can predict, eventually you'll be able to prevent. Most of the stories aren't as good as the PKU story, where it's a simple intervention. So I think we're going to move to polygenic scores and predict things like heart attacks. And that gets into the problems you're raising about IVF. So testing at birth is almost international now. So that's not an issue. We are doing genetic testing. But most of the disorders are recessive, and there's maybe 10,000 of them. Most of them quite rare. None of them are common because that's how they survive natural selection. Because for a recessive disorder, you need 2 doses. You need both alleles, the allele from your mother and the allele of your father, to have the mutation. If you only have the mutation from your mother or your father, you just have one copy, you won't show the disorder. So you can figure out the probabilities here, but you can at least see that there's many times more, 10, 20, 30 times more people walking around as carriers than there are people who have the disorder. And the way most people find out that they have a single gene recessive disorder is because they have a kid who has the disorder. Like cystic fibrosis is something people know about. Hemophilia is another. A lot of these blood disorders involve single gene mutations. So once you find out you have a kid with the disorder, you know both you and your partner are carriers. Now that's a very expensive, bad way to find out you're carriers. But once you do, then you still have the option of in vitro fertilization where you could select. In vitro fertilization, I'm sure your listeners probably know the gist of it, but you get like 8 to 12 embryos. And you can only implant one in the UK because it's too successful. It's more successful than natural intercourse. So you can only implant one. Which one are you going to implant? You normally pick the healthy one. But in this case, where the parents are carriers for a disorder, you can check the embryos to see if they have that mutation. Because can you see 25% of the offspring will be affected, 50% will be carriers, and 1% won't have the allele at all. It's just simple probabilities. So you can select the embryo that doesn't have it, the disorder. And so that's been going on for quite a long time. But now we're getting into this issue that you're raising, which is much more problematic. And that is the old issue of designer babies, but you can do it now. So if instead of doing in vitro because you're worried about a single gene recessive disorder, you could do, as Elon Musk has done, have women have many embryos. You can hyperovulate them so you get a lot of embryos, and you can pick the embryos that you think are the best. And parents define largely in terms of intelligence is that one of the big factors there. And you can predict variance in intelligence. Again, it's just probabilistic. You're predicting maybe 10% of the variance. But, in this high stakes race, people are willing to do a lot to get a little bit of an edge for their kid. So that company that does it, called Orchid, ORCID, is I think it's orchid like CHID. Anyway, it's called Orchid. They're in Beverly Hills. It's a fertility clinic in Beverly Hills, and so it's not just going to be Elon Musk. It's going to be very wealthy A-lister type people doing it, and so eventually, then other people are going to accept it. 20 years ago, when people first suggested this, designer babies, people were outraged by the possibility. But I think it's really it is happening now, and you can do it for maybe 50 grand or 100,000 pounds. It depends how many embryos you screen for example, because it is pretty high tech. You've got to, for each of these embryos, then you've got to get a cell or two without damaging the embryo, and then do the genome testing on that embryo, and then select embryos. So I think that is happening, and it is scary. There's a lot to think about there. But the other side of it is, if you did IVF and you get 8 to 12 embryos, this is a classic philosophy dilemma that's posed to students. You select say if there's 12, you select out the 3 that have a double dose of the recessive disorder. Okay, but now you have 9. Would you ignore information? Suppose one of them has some other mutation that's very damaging. Would you ignore that? Well, you wouldn't think so, but some people say yes, they don't want to choose. And that's fine. And you don't choose, you could choose the sex. You're not legally supposed to, but you could, obviously. But not to choose is to choose, in a way, right? And so you could then say, well, yes, I'd like to screen for other genetic mutations, particularly very bad ones, for example. But then you could say, what about intelligence? Would you not want to take an embryo that has a higher polygenic score for intelligence? And that's like anathema to people. But consider the dilemma you would be faced with in that situation. You can choose not to choose, but people will choose to take the embryo with the higher polygenic score for IQ, often for height. Musk seems hung up on height as well. So it certainly raises a lot of issues here, but I do think people ought to know that it is happening. And that's the bad side of it. And we got to talk about the film Gattaca from 1967 at some point here. But before we do, I would like to turn this around and say why I think it's great. I think every couple ought to do DNA testing before they have kids. And the bottom line is, finding out your carrier for a bad recessive mutation by having a kid with the disorder is not a good way of figuring out you're a carrier. Instead, you could be tested for, say, 1,000 of the most common single gene recessive disorders. And you could do whole genome sequencing so that you could look for every single gene recessive disorder and say, each of us have maybe 4 or 5 of these bad ones, but we're carriers. Our partner is going to have 4 or 5. The chances that we have the same one are pretty low, but not that low. It might be on the order of 1%, something like that. But if you could have couples screen for that, and you find out you and your carrier, your partner are both carriers, say, for cystic fibrosis. Now, that's not a great disorder. It's not as bad as a lot of them because, you live decades, but it isn't fun. So you could decide, you got options. You could do in vitro and select for embryos that don't have it, or you could adopt. You could not have kids. Increasingly people aren't having kids. Or you could say, if you're young enough, lots of fish in the sea, maybe we better try again. So there are these options. And so I would really encourage people thinking about having kids to do this. And there's one company called Nucleus Genomics in New York that I think is a very reputable company. And they do whole genome sequencing, and they offer a 2-for-1. So if you get it, they offer you a free one for someone you're thinking about having children with, because they feel, as I do, that testing for carriers is such an important thing to do. And there's some really concrete examples where it's made a huge difference in terms of cutting down on the number of these single gene recessive disorders, which is the vast majority of the major genetic problems that we have. So that's my little soapbox pitch there. But it's not all bad, is what I wanted to say.
Do you think there will be societal pushback? So maybe in Europe, I could see it being more widely accepted, but in the US, because of the political situation, because of the political divide, do you think that there'll be accusations of scientists playing God, or that it's unethical from a religious point of view?
Well, I don't do religion, but on the politics side of it, and I think if you want to talk religion, I was raised a Catholic. I became an atheist at about age 10 for reasons I could talk about. But the Catholic Church has it right. The only ethically principled position here is to say, do not mess with fertility. No contraception, no abortion, no nothing. That's the only kind of consistent point of view, because once you start doing anything, like even contraception, let alone abortion or any of these other things we've been talking about, it's a slippery slope. And you say, where does it stop? And I think ethically, it's very difficult, because there's three things to consider. One is society, the other is the parents, and the other is the kid. Each of them has rights that we have to respect. And as parents, you could say, I don't care what my kids like, no matter what problems they have. But if you then have a child who needs institutional care the rest of their life, and society has to pay for that, I think society is right in saying, well, wait a minute now, you're not making a decision just for yourself. And then similarly, there's the rights of the kid, including, you could say, the rights of the unborn kid, but that's not an argument I buy very much, because you think of almost all the eggs and sperm don't make it anyway. So you can't say we're going to have every single egg fertilized, for example. But I sort of don't want to get into those sorts of issues, because that's so far beyond my pay grade. But from a scientific point of view, this is happening, and some good could come out of it, like carrier testing. And I also think that the technology is there. So right now, very wealthy people like Musk are taking advantage of it.
And I guess we should say as well that with these complex traits, it's not deterministic, but you're maybe reducing the risk. It's not necessarily a guaranteed outcome, but it does give you the ability to maybe, like you said, increase the chances of having a taller child, or maybe a more intelligent child, or maybe reduce the risk of a complex disorder like schizophrenia. Is that correct?
Yeah, that's right. Definitely. I mean, this has only been going on for a decade, really. So it's a tremendous advance in knowledge for a short period of time. And I think most parents would, if they're making this choice about which embryo to implant, would look first at medical things. Like, wouldn't it make sense to look for cardiovascular disease? I mean, if you're going to choose at all, and again, the only ethical principle here is you don't do any of this stuff, the Catholic Church sort of approach to it. But if you've done IVF, you've got to be considering that, you're going to select one embryo. You're not going to implant 12 embryos for sure. So you've got to select. And would you not want to know if I told you, well, these two embryos have very high risks for cardiovascular disease or for some other medical disorder? And it's difficult because, say, you only have 6 or 8 embryos to choose from. It's like you got to do this odious thing of a menu. Like, do you value intelligence more than medical risk for diabetes? Or, it's really an odious sort of task in some ways. But, I think for me, knowledge is power, and I would always rather know than not know. And even if the end, I have to flip a coin, because I say, I don't know, how do I balance, this disease against that disease or whatever? I guess I would still like to know. So, but, I think it's the fact that these findings have led to these difficult issues in a way is good. The problem with a lot of psychological research is it doesn't matter. No one will ever get upset about it because it doesn't matter. These findings matter. And because heritability is such a big factor in influencing who we are, probabilistically, I will emphasize. So, it's a great question about the use of prenatal genetic testing, which is the big one. And we could at this point talk about Gattaca, this movie.
So actually, I was first introduced to this when I think I heard a podcast with you and Sam Harris. It's probably 5 years ago now.
Yeah.
And I watched it after. So do you want to talk a bit about that? And maybe will you say as well what it got right? Because obviously this was before, I think it was before the human genome was sequenced.
Absolutely, 1997. And the human genome wasn't sequenced till 2003. They were working on it in the 1990s, but the film is amazing in terms of its prescience. It got everything right. Now, when people decided to do the whole genome sequence, 3 billion base pairs of DNA, it took a decade of 200 and some researchers around the world and $2-3 billion to do it. It was a huge job. It was blue skies research. We can do it. Let's do it. Good things are bound to come out of it, scientifically at least. But no one anticipated what would actually come out of it. Like for us, the cool thing is there is no human genome. It quickly became apparent we have millions of these DNA differences. So we could then study those differences to ask if they make a difference in behavior. But Gattaca got the 4 major advances right. And one is DNA identification. In the film, I won't go into the details. It's a very clever film, though, where the reason it's called Gattaca, what are those 4 letters? Those are the 4 letters of the DNA code, G, A, T's, and C, Gattaca. And it's an academy for high flyers, literally, who want to be astronauts. But to get in this society, it's all based on DNA. And Ethan Hawke wants to be an astronaut, but at birth, he had a very high polygenic score for cardiovascular disease, early cardiovascular disease. But the subtitle of the film is "There's no gene for the human spirit." So the thrust of the story is that Ethan Hawke uses false DNA from Jude Law, who is this super human being with high scores on everything, who broke his back doing athletics, so he couldn't do any of those things. So Ethan Hawke subverts the DNA identification approaches in urine, saliva, hair follicles, using Jude Law's DNA as his. So he fakes out the system. And DNA identification, though, is so valuable. That's what has allowed, I think, 24 people to get off of death row in the US, because there were DNA samples, but this guy's been incarcerated for decades and they couldn't do anything with the DNA. But now you can say, absolutely, you, the prosecutor, say that's the DNA of the murderer. Here's the DNA of this guy. It's not the same DNA. This cannot be this guy's DNA. So that's allowed them to get off. And the other major advantage of DNA identification is to identify bodies that have been dead for a long time, most notably, say, with the Romanovs, and the Russian Revolution killing many of them. And Prince Charles, now King Charles, had to contributed his DNA because it was informative in terms of the Romanov family. So DNA identification was a very cool thing. And it's kind of like the ultimate identification, much better than the biometrics we use, like eyes, that they use at the airport these days. So the second advance, though, is something that was very popular on websites just a few years ago, DNA dating, where these companies came up and said, we'll tell you, if you and your partner give us DNA, we'll tell you if you're matched. They greatly overpromised. And so I think FDA's taken them off, at least in the US, because they were overpromising what they can do. But in the film, the love interest here is Uma Thurman. And she is suspicious about Ethan Hawke. And so she surreptitiously finds a hair in his desk drawer, because hair follicles, not the hair, but the bulb at the bottom of the hair, if you pluck out a hair, you get the whole follicle, that has DNA. And so she found this hair, she takes it to a DNA dating service, and they give her a readout. And the readout is just fantastic. Phenomenal athlete, incredibly bright, everything perfect. But that was Jude Law, of course. And Ethan Hawke had anticipated this and gotten a hair from Jude Law and planted hairs around so that if people were looking for his hair, they'd find hair, but it wouldn't be his. So that's DNA dating, though, sounds frivolous, and it is sort of better than just...
Do we kind of do that anyway, though, because of assortative mating?
Exactly right.
Do we not naturally?
Exactly right. People say,
DNA dating, so we don't have to help us.
Yeah. And, people who say, what's intelligence? It's only what intelligence test measures. Nonsense. The correlation between spouses for verbal intelligence is at least 0.5. Meaning, now you look at spouses and you say, well, they're similar in height, aren't they? You don't often see a really tall guy with a really short girl, except in the case of my son. And so you see there's a correlation for height. That's only 0.2. The correlation for verbal intelligence is 0.5, maybe 0.6. And that's because the key factor there is vocabulary. And when you meet someone in a singles bar, within 30 seconds, you get a sense of their verbal ability. I mean, if all they can say is, they can't put a sentence together, you really are measuring that, even if you don't think it exists. So assortative mating is a big thing, and that's like eugenic in a way, right? We're messing with breeding. We're selecting people who are like us, right?
Yeah, it does. Do you think it exacerbates or facilitates the continuation of inequalities and stratification of society? Because if you look at interventions over time, they don't always work the way you think they would. So is it possible that assortative mating is one factor in long-term inequality? Because I think it probably is, because it's hard to get away from that, isn't it? Like it's right there, and it's really not talked about enough. It's not discussed enough because people think that meeting somebody, a partner, is random, or maybe they think that opposites attract kind of thing, but that's just not true at all. So it must be a factor in long-term inequality.
Yeah, no, it definitely is. And it's part of the genetic architecture of our society, I think. You could avoid it by a totalitarian government coming along and saying, you're going to mate randomly, which doesn't mean promiscuously. It means we're just going to pick someone at random from the population for you to mate. Now, you might say, well, maybe arranged marriages are like that, but actually arranged marriages create more assortative mating because they're not blinded by romantic love, which is, it still overcomes the sensible part of us sometimes. They're looking at things like how well they're suited for each other, in good arranged marriages. And it kind of works. I think there's no evidence that arranged marriages lead to divorce more than other marriages. But outside of arranged marriages, we're still arranging our own marriages by finding people who are similar to us genetically. And that doesn't just include intelligence. So we are really doing something that would be called eugenic, which is such a bad word. But when Galton invented it in the early 1900s, it means good genetics. It's like doing good things. And so that's a huge topic. And I know just mentioning the word really pisses a lot of people off. But it's important to recognize with assortative mating, it's happening already. And the final point is, what are you going to do about it? You could force people to mate randomly. Well, you're not going to do that. Good luck with that. So it is part of our society, and it's part of the genetic structure. And as you say, it does lead to inequality in the sense that assortative mating spreads out the genetic distribution. So if you're bright with high polygenic score for IQ, you're likely to mate with someone who's also very high. And that means your offspring have like a double dose of genes for high intelligence.
And you mentioned IQ, and earlier you actually mentioned genetic trade-offs, I think, when we were talking about embryo screening. I actually want to just mention this and get your views on it. So again, G, like psychometric G, like general intelligence, has always been viewed as a monolith, and it's one of the most replicated findings in psychology, and it's clearly extremely predictive and an extremely useful finding. But over the last few years, I've seen a few genetics or genomic studies finding that intelligence can be not necessarily split, but you do have, I guess, specific abilities that can be heritable. So for instance, one counterintuitive finding here is that maybe something like acquired knowledge is more heritable than, I think it might be processing speed, which seems really counterintuitive because you think, well, acquired knowledge is what you learn in school. And like, obviously the traditional way of looking at G would have viewed that a little bit differently. So it would have almost viewed intelligence as an emergent property of all these things coming together, and like crystallized intelligence was something that you learned in school, and fluid intelligence was really where G was at, like it was the heart of G, like working memory. But is behavioral genetics actually challenging, maybe not challenging it, but nuancing it a little bit?
Yeah, actually it's a major part of my research is focusing on what's specific. So if you take diverse cognitive tests, like we do on an IQ test, there's like 10 or 12 tests that include nonverbal things as well as verbal things. You find that they all correlate positively, and what's in common among them explains 40% of the variance. That's what we mean by G. Spearman first coined used the letter G to refer to general cognitive ability, because he noticed there's this positive manifold, positive correlations among all tests. So you might think you're good at verbal and you're not good at quantitative, but you are. If you're very good at verbal, you're better at quantitative than average, at least. That's the correlation between these tests. So G is important. The fact that we test as diverse as memory versus processing speed, these spatial ability, these things all correlate. So G is important. And as you say, it's one of the most predictive things we have in the behavioral science. Predictive not just of occupational success, for example, but health even, longevity even. So G is very important. And we know a lot about the genetics of it. It's highly heritable. But what I'm interested in now is in studying genetics that are specific to like spatial ability, like especially in terms of education, to STEM. I'd like to know which kids are genetically likely to do well at STEM subjects independent of G. Because we've got G nailed, but you'd like to know people who are really good at like math independent of just being high G. So that's actually a major aspect of what we're doing now, and we're doing network analysis on that. So it's not to deny G, it's just to say we've always known G accounts for maybe 40% of the variance, but that means there's a lot of variance that isn't due to G, and that's interesting too, differential patterns of strengths and weaknesses among people. So you don't get to study that though, unless you take G out, unless you control for G.
Traditionally, G was on top and everything else was below it, and they didn't matter as much, like these specific abilities. They were kind of seen as skills. But again, from what you're saying, this is changing our view of G a little bit, where again, if you control for G, these other things do actually potentially matter. So like, if I'm interpreting that correctly, that is a bit of a change from traditional ways of looking at psychometric intelligence, or am I overstating it?
Yeah, I think you're overstating it a bit, because what you're referring to is the hierarchical model of cognitive abilities, which almost everyone accepts now. So there are specific tests and variants specific to these tests, like verbal ability or reasoning tests. But they go into group factors like verbal factors or spatial factors, so there's group factors in the middle. But those factors also correlate, and that's what we mean by G. So it's a hierarchical model, not saying one level's true and the others aren't. They're all true, and they are a statistical model of how things hang together. So I think they're all important levels of analysis, and we should be interested in studying all those levels. And for me, it's like a matter of what predicts best, what predicts, say, STEM, achievement in STEM subjects, for example, is a specific interest of mine.
That's interesting, because you do see people, again, like you said, with strengths and weaknesses. there obviously is a correlation between them still. So if you are good at one specific ability, you tend to be good at the other ones as well. But you do see instances of like almost marked strengths and weaknesses where people maybe excel in one area, but do have deficits. And if you take something like dyslexia, like obviously there is something going on there where someone with dyslexia could have above average ability or maybe even exceptional ability, but have these very specific weaknesses. So I guess what you could look at there is how could you remediate on those weaknesses? Is that where the research could potentially go?
Well, dyslexia is a good example of an issue we never got to. We were talking about dimensions and disorders, right? I don't think there's anything like dyslexia. I don't think it's a disorder. There are kids who have reading problems, but I think it's quantitative, and it's easier to see in terms of reading. Kids don't just wake up one day dyslexic, and they're not born dyslexic. And it's an example where the medical model has gone nuts in a way, because if you want to have a disorder, you can't just say, well, let's call it reading problems. Give it a Greek or Latin name, dyslexia, or with math, dyscalculia, because it makes it sound like, wow, that's a real disorder, right? Etiologically distinct. But it isn't. There are no genes specific to dyslexia or math problems. And so, again, there are problems. If kids are having trouble learning to read, that's a problem, and we've got to work on it. But what's to be gained by pretending that my child has dyslexia? Doesn't. Your kid has reading problems, and let's do something about it. But let's not pretend it's a disorder. So I think that's one issue. And the other issue is the one we didn't get into with disorders and dimensions, and that is comorbidity. So G explains a lot of these problems. But if you want to study reading problems, you'd be better off doing that specific to reading problems. That is, control for G and look at what's specific for reading. Because G accounts for 40% of the variance. Reading tests, language tests, math tests, they all correlate with each other. So we didn't get into that with psychopathology, but one of the big findings in psychopathology now is these disorders that are thought to be so different etiologically, like schizophrenia and bipolar, turns out the genetic correlation between them is like 0.6, meaning the genes that affect one are substantially the same genes that affect the other. And that's led to this topic of not G, but P, little p, for general psychopathology, just like G is general cognitive ability.
Our anxiety and depression correlate 0.8, so they're almost overlapping.
Yeah, from quantitative genetics, anxiety and depression at a quantitative level correlate 0.9. I mean, they are the same thing genetically.
The issue with these like categories that I didn't mention is like you could have, say, a symptom, you could have anxiety symptoms and you could have depression symptoms and you maybe could have concentration symptoms and could have insomnia. So you could like have these symptoms from different categories. So they don't fit into these neat like biological essentialist kind of categories. So like the problem is with the categorical model of looking at disorders, you're not accounting for this comorbidity. It just doesn't allow for easy understanding because you just have to give someone another diagnosis and then maybe another diagnosis. Maybe we should look at a network model or a transdiagnostic model, or like what you said there, the general psychopathological factor model would be a better way of actually looking at it, because you might not need to treat a disorder. You might have to treat a symptom or a cluster of symptoms, which might actually overlap from different disorders. Mm-hmm. Am I correct in interpreting that?
Yeah, sure. In fact, empirical data backs you up. I think it's something like 50% of the people with one disorder get diagnosed with another disorder. And then it's 50% of those get diagnosed with another disorder, So there's a lot of comorbidity out there. But for me, it's this Alice in Wonderland feeling like, who says these are disorders? Yeah, and there's lots of problems and lots of people have lots of problems. The idea of P, general psychopathology, suggests that someone with some of these problems are likely to have more of these problems. So it really does suggest that we need to rethink it's called nosology, classification of diagnostic problems in psychopathology, in terms of genetic architecture, because these two findings are huge. There are no disorders to begin with, they're dimensions. And second, we think of these different disorders, but genetically, they're very highly correlated. And, so there's really a tremendous gulf between what we think psychopathology is about and what genetics says it's about. And the difference is genetics is totally empirical. Whereas if you look at the history of all our diagnostic categories, it's just historical and it's based on symptoms. There's not a single disorder in psychiatry that has been etiologically based. So you look at autism, for example, and who says there's such a thing as autism? The simple answer is Kanner in 1945. He said he had some kids who had these three types of problems: communicative problems, these rigidity problems, and sort of social communication problems. Repetitive
communication.
Yeah. And, clearly kids with one problem won't be as bad as kids with two, who won't be as bad as kids with three. But the fact that he could find a few kids, and it was very rare at the time, that had all three of these problems, doesn't mean it's a disorder. It doesn't mean it's a syndrome. It just means these kids have three problems. So I just think it's all kind of, once you start thinking this way, you realize, God, this is really holding back psychiatric research, our diagnostic categories.
Yeah, big time. I think we'll have to finish soon because I think they're going to throw me out of the studio. But maybe just one or two more questions. So you see people who do have, I guess, ASD or autistic-like traits, obviously high functioning, like Elon Musk, and a few others I could think of as well. Is there a trade-off here where I think there is? I think you're going to tell me there is because I've seen some of the research where maybe having like a high, maybe polygenic score for certain disorders actually affords benefits maybe to a certain extent.
No, exactly right.
Is that fair?
Exactly right. Like I have a high polygenic score for schizophrenia. I'm at the 85th percentile for my polygenic score for schizophrenia, which sounds really bad, right? But I mean, only 1% of the population is diagnosed, so I'm not at the 99th percentile level. But what's exactly along the lines of what you're saying is, by studying these things dimensionally, we'll learn a lot more about them. And the one thing that interests me personally here is my 85th percentile score for schizophrenia. There's now 3 national studies, mostly in Scandinavia, that show that people in more creative professions have higher polygenic scores for schizophrenia. It doesn't mean they're schizophrenic. It probably means that they're creative because they think outside the box. So it could be that a high score for schizophrenia is actually a good thing, but you think too far outside the box and you're going to lose touch with reality.
Yeah, that's such a good point. I always find that fascinating. To touch, again, I need to finish soon, but just to touch on one more point. Could you just explain to me, because I don't think I understand it, why is acquired knowledge more heritable than something like reaction time?
It's because what we assume to be heritable has no relationship to what's assessed as heritable. Like if you look at the common medical disorders and you list breast cancer, ulcers, for example, people will invariably say breast cancer is highly heritable, ulcers very little heritable. It's just the other way around. Ulcers are one of the most heritable common disorders. Breast cancer and all cancers as a whole are hardly heritable. And with breast cancer, you ask people, why do you think it's highly heritable? Because Angelina Jolie. But those 2 BRCA1 and 2 mutations are very rare. So some GPs studying breast cancer never see a patient with that. They're really rare, and they're very extreme in everything. So our assumption that breast cancer is heritable and ulcers are not, you ask people, why are ulcers not heritable? Well, they say, it's just due to stress. But it's not. It's highly heritable. It's one of the most heritable medical disorders. So you can't assume what's heritable and what isn't. And to give you an example, in cognition, you ask people about different cognitive tests, they'd say processing speed, as you suggested, maybe should be highly heritable because it seems more basic, biological, right? It's not nearly as heritable. And one of the most heritable tests, vocabulary, the completely acquired thing. You're not born with vocabulary words. But to close, just to say, think about genetics. Why might vocabulary be highly heritable? You don't inherit the words, but you see people who are interested in nuances of words. They want to use a word properly. It's not like, whatever, you know what I mean, right? They want to learn what words mean what. And so they're going to develop better vocabularies. And I think it's easy to think about how genetics might influence that.
Amazing. Thank you so much. There's a lot I didn't get into. So maybe another time I could talk to you again. We could talk more. There's philosophical points and stuff I wanted to get into. So maybe another time, but I really appreciate it. It was a brilliant discussion for me. And thank you so much.
Thank you very much.
I've been a long time admirer.
Yeah.
So your work has been fantastic.
Well, thank you.
Yeah, I'm basically self-taught in this area from your work. So thank you.
Good. And good luck with your podcast. I would like to talk to you again because I know we haven't gotten through your list of questions, and there were really great questions. I appreciate you putting the effort into thinking about that. So until the next time, all right?
Thank you, Robert.
Okay.
Next time, talk again.
Bye.
Bye-bye.